So I'm Stephanie Hicks. I'm in the Blackshear lab at the National Institute of Environmental Health Sciences. And today I'm going to share the work we've been doing on zinc finger domain containing protein that belongs to the tris tetraproline family of proteins. And specifically, I'm going to talk to you about the mechanism of RNA recognition and binding. So the tris tetraproline family of proteins regulate inflammatory responses in the body. And they do this through this highly conserved zinc finger domain. And it's composed of actually two zinc fingers that have the characteristic CCCH motif that's required for coordinating zinc. So these two fingers are connected by 18 amino acids. And it's this zinc finger that recognizes and binds to AU-rich RNA, so single-stranded RNA. And so specifically, when the zinc finger binds to AU-rich RNA, it's found in the 3' untranslated region of mRNA targets. It then allows this extreme C-terminal domain, this NOT1 binding domain, it recruits deadenylases to the mRNA target and then facilitates mRNA decay in cells. So we're really interested in this protein, but specifically in understanding the mechanism of RNA recognition and binding. And I mentioned that while these are involved in a lot of inflammatory processes, not a lot is known structurally. So that's where SAXS comes in for us. So specifically with SAXS, we wanted to address the question, does RNA binding cause a conformational change in the protein? So first, we chose to look at just the zinc finger domain alone. So if you remember, it's actually two fingers that are connected by this linker. And so we use the mail-in SAXS. I mailed in a plate for high throughput SAXS that contained the zinc finger domain alone, but then also the protein in complex with the single-stranded 9-mer RNA shown here. So these are the scattering profiles shown on the left at 3 different concentrations. And then you see in the center, this is the low Q region or the Guignay region of the plot where we can extract RG values. And what we noticed immediately is that the zinc finger domain, when it's bound to RNA, is actually smaller in terms of RG than the RNA-free protein. And then if you look here on the right, these pair distance distribution plots where we can get the maximum dimension, we notice that, again, the RNA bound protein is smaller in terms of dimension, maximum diameter, compared to the RNA free form. So what the SAXS data is telling us is that the zinc finger domain is actually more extended in the absence of RNA and adopts a more compact structure upon binding RNA. And then because of this, we found this really interesting. We wanted to take this a step further. So we used another in-solution technique, nuclear magnetic resonance spectroscopy. And we actually were monitoring the dynamics of this zinc finger domain in solution. And so what you're looking at here on the left is the zinc finger domain and the RNA-free form of the protein. And any residue that is moving in solution is highlighted. So for example, zinc finger 1, in the absence of RNA, you really only see one residue moving in solution. It's here. But then if you compare the second zinc finger 2, in the absence of RNA, almost the entire domain is moving in solution. And then if you look over on the right, we found that once you bind RNA to the zinc finger, the protein is relatively static now. It sort of locks it into a single conformation. And so together, our SAXS data and our dynamics data is saying that, suggesting that this zinc finger domain is really, it's exploring a lot of conformational space in the absence of RNA. And we think that may be consistent with SAXS in terms of that we see that our RNA-free form of this protein has a larger RG and Dmax value. And so I mentioned we're interested in the mechanism of RNA binding and recognition, but for this protein to carry out its actual function in the cell, you need the full-length protein, and specifically the C-terminal domain. So we wanted to use SAXS to address the second part of this question. Does RNA binding to the zinc finger domain, again, the same 9-mer sequence to the zinc finger domain within this protein, does it translate into an overall conformational change in the context of the full-length protein? So again, we used mail-in SAXS, and I sent samples for the full-length protein and the full-length RNA complex. These are the scattering curves at 3 different concentrations. And then here are the plots from the Guignay region where we can calculate the RG values. And again, we see now with the full-length protein that the RNA-bound form is again smaller in terms of Rg compared to the RNA-free form. And then interestingly, if you look on the right, these paired distance distribution plots, we notice a few things. First, the full-length protein looks a lot different in terms of the shape of the curves compared to the zinc finger domain alone. But then interestingly, if you look at the Dmax values that we can get from these, the full-length protein has a Dmax of around 64 when it's bound to RNA, again being smaller compared to the free form of the protein, which has a Dmax of 80. And then if you remember the Dmax for the zinc finger domain alone, it was around 63. And I find this quite striking because we doubled the molecular weight of the protein, but the maximum dimension is about the same as just the zinc finger domain alone. So together, these SAXS data are suggesting that this full-length protein is actually more extended in the absence of ligand. And then when you have RNA binding just to this zinc finger domain, it actually translates to a conformational change in the entire protein to make it more compact. And so what we've been able to do using SAXS is to show that, in our case, RNA binding or ligand binding to this RNA binding domain, the tandem zinc finger, takes the structure of the zinc finger domain from a very extended conformation to a more compact conformation. And then by combining our SAXS data with our dynamics data, where we see zinc finger 2 exploring a lot of conformational space, we think that these are really complement one another and give us a lot of insight into maybe that the Rg and Dmax values that we're seeing, these larger values for the RNA-free form of the protein, may be due to this sampling of conformational space by this second zinc finger domain, compared to when you have RNA coming in and binding and really locking this zinc finger domain into a final single structure. And then some of our next steps, we now want to go into calculating molecular envelopes for the RNA-free and the RNA-bound forms of these proteins. And we want to get an idea of the overall shape. But in addition, we're going after the high resolution structures using NMR. And so we'd like to use our SAXS data and these molecular envelopes to really validate and complement our NMR studies. And then finally, one of the things that I should have pointed out is that SAXS has really been advantageous for us because we've been able to study not only the zinc finger domain, which has secondary structure predicted, but also our full-length protein, which has very little secondary structure predicted outside of that zinc finger domain. So, and then I've shown you some data that RNA binding to this protein really changes the conformation, the overall conformation in the context of the full-length proteins. So now we'd like to use some of the modeling algorithms out there to predict conformations of the RNA-free and bound form of the full-length protein. And then all of the data that I shared with you today is on the smallest family member. So it's only 100 amino acids. However, most of the family members contain up to 300 amino acids, so they're much larger. So I'd like to use SAXS to evaluate if this mechanism we're seeing with one of the smallest family members is conserved across the family. So does RNA binding to just the zinc finger domain cause an overall global conformation to these proteins? Is that a mechanism, a conserved mechanism? And with that, I will take questions and input. And I'd also like to convey that we are in the process of writing up a manuscript and we'd like to include the SAXS data. So any input you have would be extremely valuable to us.
Great. Those data, Stephanie, what you show are the high throughput data or SEC
Yes,
SAXS?
they are high throughut. I did try SEC SAXS actually, and we got some good data, except I didn't get great data with the zinc finger domain alone. And I realized yesterday from Rob's talk, so one of the purification steps that I use is a heparin column. And so I think that, yes, it bound to the, so I sent like 5 mg per mil concentration and I ended up with no signal. And I sent 2 different samples and I think that's what happened.
No, those data that you showed are pretty good enough. They don't really necessary to run SEC SAXS. I mean, that's my personal opinion. They're really good.
Okay.
And also, it is very small protein. You mentioned a couple of 100 amino acids. Yes. So if you're gonna considering some very simple ab initio shaping, and that's just my personal opinion, Rob will probably disagree, and Greg as well, but it will be just a blob. it will be not a lot of information. You have all the information in the raw function. You can also plot a Prati plot for the difference in dynamicity or flexibility.
Mhm.
But you already see everything with the raw functions. These are histogram of the distances. For the small protein, it's good enough. But if you really want to extend some modeling, for example, for the full length protein, we really recommend to use a simple rigid body model, just atomistic models, because you know zinc figures are zinc figures, linker in between, and some double-stranded DNA, and just put that together. You remember the presentation from Jimmy Vale? It's exactly what you probably will end up. You don't have a structure, but it's probably very, very similar stuff.
Okay, great. Thank you.